Okay, let's talk about the protists. So about 10 years ago, or maybe about then, the Protista were one kingdom. But with advances in biotechnology, they started to sequence DNA and started to go, and these are like so different. There are so many different kinds of protists. So they are working on kingdoms right now. I know I mentioned this, that the protist scientists got together and said, let's scrap this whole kingdom, phylum, class, order, family, genus, species, and let's start to talk about supergroups and divide that way. So there will be, what this is going to be in terms of the taxonomy will change a lot in the next 10 years too. As I mentioned, I like to mention career paths. If it's something you're interested in, if you're interested in biotechnology, or you're interested in biomedical engineering, or bioengineering, I should say, there's so much here, especially with the protists as well as other organisms, where you can start to sequence DNA and compare, look at evolutionary trends, and start to help classify things. So lots of interesting stuff going on. Okay, so some of the reasons why they are so diverse is that most of them are unicellular, but there are some that are multicellular. And then there's these ones in the middle. The colonial ones are very interesting in that they are a group of unicellulars that live in a group together, like roommates, and then they divvy up the jobs amongst themselves in a colony. That's why it's called colonial. They live in a group, a colony. And they function like a multicellular organism, but they're still unicellular organisms. So this is kind of a brilliant in-between thing. We have seen over time that these colonial organisms have evolved to become multicellular organisms through the example of the endosymbiont theory. I know we've talked about that before. In terms of the mode of nutrition, some are autotrophs. This term autotroph means self-feeder. It's not like you and I who, you're a big person, you can feed yourself. That's not what that means. Biologically, autotroph means you can feed yourself inside of your cells through the process of things like photosynthesis or chemiosynthesis. So this is a huge advantage. You don't have to rely on other organisms so directly for your energy needs. And then heterotroph, hetero means other, troph means feeder. You have to feed on something other or outside of your own cells. And then reproduction-wise, most of them reproduce asexually, just like what you saw with fungus and the budding. That's asexual reproduction, where they start to form that bud out of the side of themselves. It makes a genetically identical copy of themselves. That's one example of asexual reproduction. Our mitosis is a form of asexual reproduction where the cell grows to twice its size, replicates two sets of genetic material, organelles, and then splits into two cells. That's another form of asexual reproduction. But there are still many that reproduce sexually, and that involves sperm and egg production. So there's just and these are some of the just basic things, which shows you a great diversity amongst the protists, and we'll get into some other things soon. So just some pictures of what they look like. So we're going to start out with, so like here, you can see this one, for example, has green dots in it. This indicates that this protist is an autotroph, that they undergo photosynthesis, that they have chloroplasts inside of them, and the chloroplasts undergo photosynthesis. They have chlorophyll. So let's categorize our protists into plant-like, fungus-like, and animal-like protists, just to give us some ideas of, again, another form of diversity within the protists. So the plant-like protists, these are autotrophs. They make their own energy inside of their cells through photosynthesis. These are called diatoms. Diatoms are a form of phytoplankton. Photo. There's a photosynthesis component. So again, they're autotrophs. The term plankton refers to water environments in which an organism cannot direct where they're going when a current comes. So anything that is plankton, if a wave comes or a current, they can try and like move as much as they can, but they're going to go with the current. The phytoplankton are the ones that photosynthesize in that area of the plankton. We also have zooplankton or zooplankton, and those are animal-like organisms that go with the current. So the phytoplankton here, these diatoms are a form of algae or seaweed. Sometimes it goes by one or the other name. Diatoms are unicellular and they are microscopic. So here we have an algae or a seaweed that is microscopic in nature. It's not like when you think about maybe algae or seaweed, you might think of like green, gooey, kind of plant-like stuff in water. These are very different. So here you have a picture of them. They are beautiful shapes. They have a glass-like outer covering called silica. Oh, so before I get to that. So again, they're autotrophic, self-troph feeder. They feed themselves inside of their cells through photosynthesis. And like algae you might picture in your head, they are a little bit different in that they are single or unicellular organisms.
Would that be considered microscopic?
Yes, that is a picture from under the microscope. Yeah, good question. Yeah, so this is a microscopic blown up image. So when you're swimming in water, you're at the top of the ocean, there are billions and billions and billions of these floating around in the water with you. They're all around you. This is amazing. Those algae make up 70% of the photosynthetic activity on the Earth, at least, and even some figures say 80%. So thinking about like the oceans are vast. Your connection to the ocean is direct. Even though you might think like, well, I don't live close to the ocean. I don't have a connection. So what if the oceans are through climate change? What if they're acidifying and overheating? Here's your direct connection. The air, the oxygen in the air that we breathe, 70% of that oxygen comes from the oceans. So when we're talking about the effects of climate change and acidification, which will have a direct effect on acid having a negative effect on the growth and development of those little microscopic organisms that photosynthesize, that's a huge deal. Just like our bodies, right? If we have too much acid in our body, we have this thing called homeostasis, that in our bodies, for general, our cells like to remain about neutral. When our cells get too acidic, we can die. When they get too basic, we can die. We have specialized cells, like our stomach, for example, works in a good acidic environment, but most of our cells, they want to maintain their homeostasis. It's just like the microscopic diatoms, they want to live in a fairly kind of neutral environment. So with climate change, the absorption of carbon dioxide into the ocean actually causes that carbon dioxide to change into an acid, and it acidifies the ocean water, which is going to directly affect them, which is a producer that almost every organism on the earth relies on in terms of the oxygen that we're breathing. In addition, warming. We know that carbon dioxide also causes increased warming activity on the earth. We like to maintain our body temperature around 98.6 degrees. Same thing with them. They like to maintain a warm environment, but not too warm and not too cold. And so with acidifying and warming of ocean waters, it's going to have a direct effect on this, and then that's going to have a direct effect on everything in the world. The other thing is that they are the basis of the marine food webs as well. So that when you have these diatoms, and they are food for the phytoplankton, are food for the zooplankton, and the zooplankton are food for small or juvenile creatures, and then the small juvenile creatures are food for bigger, larger creatures, and that affects the whole system as well. So we have a lot of connections between if this falls, a lot of things are going to fall apart. Let's talk about another kind of algae. So this might be what you would picture in your head, more kind of like algae. These are tall seaweeds or bigger seaweeds, multicellular. We can see them with our eyes rather than the diatoms who are phytoplankton. This, for example, here is a kind of algae that lives in cold waters. So think about the west coast of the United States, California and Oregon and Washington. This is the base of those ocean food webs, brown algae. It's often called kelp. Kelp makes these underground, underwater forests. These strands can be hundreds of feet tall. So I love this picture because this shows you, it almost looks like you're looking at a forest, but underwater. And you've got these tall kinds of brown algae that can allow for homes. They can allow for hiding places, for predators, for prey. They are food. They are producing oxygen. They are the base of that entire food web. And then this is another kind of algae called red algae because it has a red color to it. Red algae is very interesting in that in oceans, they'll take calcium carbonate and they will put it underneath their skin. And this is a good source of calcium for many countries around the world who live on oceans. Red algae, because it has that calcium carbonate, like what we have in our bones, it can, when it comes apart or when it lays on structures like rocks, it can help to cement these areas like coral reefs, for example, and other rocky environments, and it makes cracks and crevices in the rocks to make homes for other organisms. All right, so how about you? Did you have any red algae today? I hope you did, because a lot of times red algae, which is listed as carrageenan in your ingredients and things like toothpaste, so hopefully we all brushed our teeth this morning, and other things like that are gooey, like chocolate milk, whipped creams, ice creams, they all have carrageenan or red algae in them. A lot of our cosmetics as well, they have carrageenan in there. It's like makes things that are, makes them a little more goopy and less runny. All right, and then this might be the one you're most familiar with is green algae. Green algae, we could walk out to the pond here or any ponds in the area. The Great Lakes will have this color algae. This kind of algae is more freshwater ecosystem related. These plants are what we believe to have a common, this algae is what we believe to have a common ancestor with the land plants. Is that if you remember back when we talked about evolution, is the first land plants, they had come from some kind of plant-like thing like this from water or from the ocean. So there's a common ancestor there. All right, other interesting things and maybe other career paths that are getting hot here is green algae is being used as a biofuel. So here's that word bio, that living things are being used to make fuel for us. These are what we would call algae farms. So right now, let me just mention about this, is that people are working really hard to try and use algae to make fuel. And they're using these farms. But remember that even like this, they look like giant test tubes, that these test tubes that are outside, or these that might be in like a big greenhouse, these like pockets of algae, that you also need a way to maybe like turn on lights. This will, like if you're inside at nighttime, keep it warm on the inside. If this is somewhere in a warm climate, you might not need that heat as much during the night or in the winter months. But if we're looking at a place like, let's say, in the middle of Illinois, that they have a farm like this, you're going to need some kind of factory-like structure that is like a greenhouse. It's got to keep it warm in the winter time. and glass will let a lot of that heat escape. So you're pumping fuel into this to grow this kind of fuel. So you need fuel to make the fuel. And even if you're looking at an outdoor algae farm, you also have to take these things and then put them through a chemical process to change them from being algae into being fuel. So you do need fuel to make fuel because you need all that processing to do it. Right now, and when I talk about the profits are not justified yet, because it's like, so let's say that your algae produces this much fuel, but it takes this much fuel to process making this fuel. So you have a negative net gain, or you have negativity here in terms of your profit. What they're looking for, brilliant minds like you, to get us to a point where there's a positive profit, that you actually use less fuel to make the fuel itself. However, let's think about the bigger picture with this, though. Our fuel, like what you put into your gas tank typically, is from fossil fuel. What we mean by fossil fuel is that fuel comes from oil, natural gas, or coal. Those things are from dead ancient ancestors in ways in the earth they've been compacted to become, from living things to become fuel over time. We only have a finite amount of them. So what I mean by that is that there's only so many ancient dead ancestors. We could run out of that. But this, even though the profits are not quite there yet, they're actually relatively close. This is renewable. We could keep growing this algae and resupplying fuel, even though we're not quite there with the profits. So this is a little bit better in terms of, yeah, it's renewable. Now, if you could get it here, you'll be even better. Interesting thing about this kind of algae, though, is the algae that they use can double their body mass in a day. So it grows quickly. Imagine you today doubled your body mass. Let's say that you weigh 150 pounds, that by the end of the night, you're 300 pounds. And then tomorrow you're 600 pounds, and then 1,200 pounds, right? So it's pretty amazing in terms of how much can be made and how quickly, that it's a very sustainable system. But again, we're looking to get that profit, just get it over. So if any of you figure that out, you'll be famous. Lots of ways to use your brilliance. There's another kind of algae called dinoflagellates. Dinoflagellates. These are photosynthetic. They're free swimmers, this kind that I'm looking at in the picture here. They're unicellular organisms that can swim on their own. This kind here, which is red in color, produces toxin as it grows. So if you've ever heard, like down in the Gulf or around Florida, that the beaches are closed from red tides, that's this kind of dinoflagellate. And the toxins that they produce can poison shellfish, so like oysters, for example, shrimps, lobsters, crabs, scallops, all the things that you might like see on a seafood menu. They could poison that, and they can have dire effects on human economy. They can put people out of business. The other thing is that, let's say you go down to Florida for a vacation and you're like, I want to be at the beach every day, I want to swim in the ocean, and then that is there. You can swim in this, but most people will get irritation on their skin from the red tides. So it's not as pretty. It's like kind of reddish brownish, so it looks yucky. There might be dead organisms floating in there, right? You don't want to go on the beach for that. So it can have an effect directly on food economy, but also on just tourism as well. And then another kind of dinoflagellate, they're called, here's that zooxanthellae. These are found and have a symbiotic relationship with coral. They're found underneath the skin of coral polyps. So the very animals that help to build coral reefs, which are the basis of tropical ecosystems. And you can see here, it's pretty cool picture where the polyp is this clear animal, and it has little zooxanthellae underneath its skin. So just like we talked about plants, and when we were looking at mycorrhiza, for example, that this organism can get photosynthesizing and I say lichen. So just like lichens, how you remember the lichens it's a fungus that has the algae or the photosynthetic bacteria that live underneath their skin. It's a very similar situation here. But here we've got an animal with an algae living underneath its skin. And this can give the animal up to 90% of its energy needs too. Now, with warming of the oceans, what happens is that the zooxanthellae are like, I'm out of here, warming and acidification. So that the coral polyps, imagine that you were given like 90% of your money every day by something to buy your food and everything that you need, and then suddenly that money source is cut off. What are you going to do? Suddenly, right? You're going to struggle a lot. Are you going to survive? Well, sometimes organisms do survive. Sometimes these polyps survive, but it's very stressful. Stressful to their survival, which means they're going to put reproduction on hold. Many of them do die. So we're seeing this effect on coral reefs directly from the warming and acidification having an effect on the zooxanthellae, and then the corals are diminishing. Next group, let's talk about the animal-like protists. These are the ones that I think are like in your nightmares, that if you could make a movie, like the very vintage scary movie, The Blob, if anybody knows that one. The Blob was this big blob that would go around and it would eat people. That was based on an animal-like protist. So amoeba are the animal-like protists. That the amoeba is that idea that you can come and just like, you can grab something with some like jelly-like arms, take them inside your body and then digest them inside of your body. That's what that was, the blob was based on. Somebody was like, oh, that creepy, scary, animal-like blob thingy, and they made a horror movie about it. So these are non-photosynthetic. They do not photosynthesize. That's heterotrophic, other feeder. You get your energy and your nutrient needs from somewhere other than your own cells. So here are some of them. Plasmodium, which causes malaria. Paramecium, you might have looked under the microscope and saw them in like 3rd grade. The zooplankton, and then amoebas, as I mentioned, the blob. So let's talk a little bit about the amoebas. Amoebas are usually in water environments, but they can also be in, when we say terrestrial, we mean land environments. See these little like nubs that are coming out? They call those nubs pseudopods. Pseudopod means fake arm. So they can actually they're like a gooey jelly thing, and the pseudopods can grow. And so they can have these pseudopods that kind of come out and engulf something and take it into their body, which makes them a predator. They are a predatory little microscopic thing. They also are parasites. If you've ever gone to a foreign country where you get that like digestive disorder, you probably drank some water that wasn't completely filtered, that's usually associated, well, not always, I should say, sometimes associated with a parasitic amoeba. Sometimes you'll hear, every so often we hear like somebody was swimming in a lake and then they died and they found a parasitic amoeba in their brain. Yeah, sometimes they are going to get into our bodies and like eat our brains too. So let's talk about Plasmodium. Plasmodium, this is a kind of animal-like protist that causes malaria. So this is interesting because here we have a parasite that has 2 different hosts. It's got a host that's a mosquito, and then it's got a host that's a human. So mosquitoes will get it by, if a mosquito, stings you, and what they're doing is they're injecting their nose, it's called a proboscis, into you. and they're sucking blood out of you. And when they're sucking your blood, what can happen is if you are infected with Plasmodium already, that they can get the Plasmodium on their nose, and then they start to carry it. And then they go and they sting somebody else, and then they give it to that other person. So they can bring it from person to person, but they get it from an infected person in the first place. When it gets into our body, let's say that you don't have malaria, and this little mosquito comes and it has the Plasmodium on it, boop, sucks your blood, basically injects it into your body, and then those Plasmodium travel through your body, and they go and they settle into the liver, and in the liver then they'll spread to your blood system, carried on the red blood cells, and they actually start to pop your red blood cells. Your red blood cells carry oxygen, so now if you're starting to pop your oxygen carrying cells, what happens is the process of using oxygen diminishes, or the ability to have oxygen diminishes. You don't have as many red blood cells to carry oxygen. Oxygen is critically important in cellular respiration for breaking down our big macromolecules into smaller molecules and getting that ATP made to fuel all the processes within our cells, our metabolic activity. And so it lowers your metabolism. The places that get most affected are like, first, you might notice, you might not know you have malaria right away, but you might get some tingling in your fingers and your toes, your nose, your ears, because those areas that have a lot of capillaries or little tiny blood vessels in there, they're going to be the last places that can, or I should say the first places that will get broken down and affected. Your liver too, you might start to get yellowish eyes, that jaundice issue. So there's some clues that you might get that. Fever is another one. When your body notices, when your immune system notices like something's under attack, your immune system goes into effect. Our body reacts to anything under attack in our body by upping our body temperature a little bit, which does two things. One, it makes our white blood cells work faster. It gives them a little boost. It also kind of simultaneously, it ups our immune system, and it lowers the ability of whatever that foreign invader is to work. So a higher temperature, they don't work as good, but our immune system works better, and you get that little bit of fever. So, when you got a fever, something's not right in your body. So this just gives you an idea of what's happening, and then you can get your liver infected. It travels to your liver, the Plasmodium, and then the Plasmodium start to break down your red blood cells. All right, last group, the fungal-like protists. A lot of these they thought were fungi, and then when DNA was starting to get run, they were like, oh wait, no, these are totally different than fungi. So the fungal-like protists, they look like this. Doesn't that look like one of the fungus from your lab the other day? Yeah, so structure-wise, I think it was pretty smart to say these are fungus because they have that same structure of looking like mycelium, and then the strands, the hyphae, and then when you have the sporangia and the little tiny spores on there, it all looks very much like a fungus. So these are classified, one group is classified as the slime molds. And they also have those filamentous bodies like the hyphae, however you want to say it. So big advancements in biotechnology then to make these different groupings based on their genetic material. And some of these you might know as many of them are decomposers. So we often say it's the bacteria and the fungus who are the decomposers, but also some protists are decomposers as well. There's always an exception to the rule. There was probably a common ancestor here with the fungus that this branch also can do decomposition. This group that can decompose, they often will be found to break down food crops like potatoes. The famous great Irish potato famine was based in this kind of protist. We do hear often like vineyards, avocado trees get attacked by these protists, and people lose a lot of money in their crops here. I think that's it. Okay, so that's it for the protists. So just a little glimpse into the diversity of the protists. So let's talk about the prokaryotes. When we are looking at the prokaryotes, we are looking at two domains, the bacteria domain as well as the archaea domain, or archaea. Again, that's another one I go back and forth on. So let's define some terms that we've been looking at. Prokaryote versus eukaryote. The term prokaryote means that the cells have no nucleus. Pro means before, karyotic, nucleus. So they evolved before cells with a nucleus evolved. Excuse me. We know that from studying evolution. We said that those prokaryotes were the first organisms that lived on Earth, and eventually 1 billion years later, we get the evolution of membranes, and then we get the evolution of the nuclear envelope with the evolution of membranes to give us a nucleus to protect the genetic material better than the genetic material being exposed within the body of the cell like a prokaryote does. Prokaryotes are much simpler in nature. They don't have as many organelles. They don't have as many structures. But does that mean anything in terms of being good at survival? No, because they've been around for billions of years. Prokaryotes have been around for about 3 billion years. If humans, we've been here for about, in our form, maybe 6 million years, and things are looking a little hairy right now for the humans, right? We could be gone, and they'll just be thriving away for probably billions and billions more years. Eukaryotic cells have a nucleus, so as I mentioned, with the evolution of membranes, one of the membranes that evolved was a membrane around the genetic material, which gave us a nuclear envelope, controlled what could come into the environment with the genetic material, could keep a lot of the stuff in the cytoplasm out, but could also let things that are really important, like mRNA, out, so that you could go from your DNA to mRNA to making proteins and other really important structures in the body of the cell. EU means true karyotic nucleus. So these are cells that have a true nucleus. They have a membrane that is protecting the genetic material. If something goes wrong inside of the cytoplasm, you have a true nucleus to protect your genetic material. Not only does the nucleus have a membrane, but all of the organelles have a double layer bound membrane. So control over not only what goes into the nucleus or out, but every organelle can control what goes in and out, what they produce, what they take in, what they package. Even the things that they package have a membrane, so that even if you are like the endoplasmic reticulum and what product you're making, you put a little envelope around it and you send it over to the next thing safely without it being exposed to anything in the cytoplasm. So we have a lot more complexity, not only in just structure of the eukaryotic cells, but a lot of complexity in the metabolism, all of the cellular reactions that happen in the cell. Again, does that mean better? You could debate that all day. So here again, here is our phylogenetic tree. We can see the taxonomy, the naming of these groups, and even here what we see is like the kingdoms of Eukarya, no kingdoms yet here. Who knows what will happen with that? But what I want you to take a look at is the majority of this tree are really these two domains here. Even though these domains taxonomically do not have names on their branches, it doesn't mean that they're any less than this. This is easier to identify for us. Also, notice that you have everywhere, remember that when we're looking at these trees, that everywhere you have a branch made, there's a common ancestor here. So when we're looking at the first organism, they're not sure. There's a lot of hypotheses that say the first organisms were archaea, but nobody knows for sure because nobody was there. We don't have that data. So they kind of do this like blending thing here, but we do know that bacteria and archaea come from a common ancestor, come from the common ancestor, and then from the archaea come the eukaryotes. Okay, so we need to talk about that. All right, so let's talk about just common shapes. There's a lot of different shapes, but three very common shapes of the prokaryotes are one, spherical, sphere, round, called cocci. So you see these ball-like shapes here. And then you have the rod-shaped that are called bacilli. And probably if you were like conjuring up bacteria in your head, you'd probably think of cocci or bacilli. A third shape, pretty interesting in its nature, very similar to what our DNA looks like. It's a helix shape or a spiral called spirilla. So they would look like this, but if we're looking at a microscope image, they're going to look kind of like snakes. So they'll be wavy. So again, you've got the cocci, the bacilla, and the spirilla. This is a better microscope slide. This is an electron scanning microscope, and you can see a little more of that spiral shape there. All right, so just based on your own knowledge of bacteria and archaea, which of these situations do you think you would find any prokaryote? Would we find them in the intestinal tract? Yeah, we've definitely got them there. Do you think you'd find them in polar ice caps? Do you think you'd find them in deep sea vents? You think you'd find them out in a mud puddle? Yeah, they're everywhere. So you are going to find them everywhere. All right, so a little bit about their reproduction. Their reproduction is called fission. The term fission means to split in two. If we could do, well, this is very similar to our mitosis, that what happens with our cells and what happens in archaea and bacteria is that they grow to twice their size, reproduce two sets of organelles, reproduce another set of genetic material, and then split and become two new cells, just like what we do with our mitosis. So here, this is an asexual form of reproduction. No need for sperm and egg. But you get an almost genetically identical daughter cell or daughter cells from your parent cell. So you don't get as much evolution, but we'll talk about that too in a second. Certainly when the parent cell is doing a quick job of copying not only the genetic material, but also all of the organelles, and then going through this process of splitting everything equally between two new cells, do you think mistakes can be made in the copying of the DNA? Always, right? So always you can get a little bit of evolution in there in just those accidental mutations that happen when you copy the genetic material. So certainly there's always an opportunity. When a textbook says 2 genetically identical daughter cells, it doesn't mean always, because you can get those mistakes. So here's why I said the but. So you could get a little bit of evolution happening just by accident and making the 2 new cells. And look at how fast they're doing it, 20 minutes. So within 20 minutes, and this varies among species, how quick their generation times are, that the quicker the generation time, the more possibility of making those mutations. And so you can get quick evolution because they reproduce really, really quickly. I mean, think about us. When we make an offspring, it takes over 9 months to do so. This is also why if in the morning you ever wake up and you're like, you feel a little scratchy and you feel like just a tiny bit run down, you feel off. And then through the course of the day, you just start to feel worse and worse and worse and worse because every 20 minutes they're boop, 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 boop. That they are exponentially growing. Like if you've had strep throat before and you start out with a little scratchy throat and then throughout the day it gets sore and more sore and more sore and more sore and then you just feel awful by the end of the day. That's because this is happening. One of the mutations that can be made, and what we have seen in our lifetimes, is that they can make mistakes or mutations that allow them to live in the presence of different antibiotics, that give them antibiotic resistance. So remember that when we talked evolution and we looked at lab 22, that this is one of the things that just by chance and accident, they evolved mutations that allowed them to survive in the presence of antibiotics and has put us in a dire position as well. Here's another weird thing that they do. They do what's called HZT, horizontal gene transfer. This is really wild. Once they have mutations, what horizontal gene transfer is telling us is that not only can they share genes amongst members of their own species, but they can shoot genes over to other species too. And so with issues of antibiotic resistance, so if we have a cocci bacteria, so this is one species, and then let's say that you've got a bacilli bacteria, that when we take a look, at their genetic material, what we know about their structure is that not only do they have a not only do they have a chromosome, bacteria have one main chromosome. It's called a nucleoid. And the nucleoid is usually attached to the inner plasma membrane, which is then attached to a cell wall. And so you see it stationary within the cell. But they also have little tiny pieces or circles of DNA that are called plasmids. Plasmids are these little circles of DNA that are free in the cytoplasm as well. Just because plasmids are tiny pieces of DNA doesn't mean that they're not important. Plasmids can also have, and do have, important genes on them as well. So, when we're looking at horizontal gene transfer, what can happen is these are clearly two different species, right? They're different shapes. So, well, I should say, I say clearly, like, I'm going to say these are different species. So by indicating they're different colors and different shapes, I'm indicating these are two different species. What can happen between different species? And we know that in studying evolution, that with evolution, we have those evolutionary mechanisms that separate us from reproducing with other species. But here, now they're not reproducing sexually, but they are sharing DNA. So this is like a little weirdness in evolution, is that they can, I shouldn't put it there. They can make these bridges. And on the bridges, what can happen is they can share plasmids from different species. So let's say that, and how and why is a big question, but if these pieces of DNA, these plasmids, have genes for resistance to different antibiotics, now each of these, by making this process of horizontal gene transfer happen, they have just upped their survivability by sharing resistance to more than one antibiotic. This is crazy, crazy stuff. So it's kind of like sexual reproduction without involving sperm and egg. It does involve DNA, but it doesn't involve sex. Crazy, right? So the evolution, what we call this process of making this bridge, is called conjugation. When you conjugate, you come together. Call it in quotes mating because it's not sperm and egg related, but it is DNA related. So kind of doing something like what we do with reproduction, but not really. In this picture, you see a bacilli, bacteria. You have two different bacilli bacteria. This one has these pili on there. It has these long kind of hair-like things on there, and this one doesn't. So these are two different species, but you can see this bridge. And what they're doing is they're doing that horizontal gene transfer right here. So cool and wild. So there's two different ways that they can do it. One is that they can extend their cell membrane out of their cell walls and make a cytoplasmic bridge. So this is kind of complicated. Because these cells have cell walls around them, they can kind of open up a pore in their cell wall, shoot out cytoplasm to make a bridge and share plasmids. So that's one method of this. The other one is that they use the pili, like you saw in that last picture, those hairs. Some of them are called sex pili or pilus. I think pilus is single, pili is plural. So they can use one of those pili extensions that pokes into the other one. So cool. Cool for them, not cool for us, but definitely cool for them. So this is another way. In addition to their quick rate of generation time and making mistakes that way, this is another way that they can make new genetic combinations amongst bacteria and archaea. Who knows? One of you may discover another way that they do it. I mean, they've been around for billions of years. They resist really harsh conditions. I mean, imagine like bacteria that go through our digestive tract. That's harsh. There's a lot going on there, and they survive that. So, who knows? All right, by what process are bacteria able to gain resistance to antibiotics? This could be a question on your exam, right? We just went through all this stuff. And would it be by forming endospores? We didn't really talk about that. Did we talk about meiosis? Conjugation? That sounds like a familiar term, right? Sexual reproduction? No. Mitosis? So here I throw in some terms that you know and some that I mentioned, but definitely the correct answer is conjugation. So easy to answer this now, but on the exam, you've got to remember this whole process of horizontal gene transfer. A follow-up question could be, what is this process done through? Or what is it called when they share genetic information? And that could be horizontal gene transfer. So just putting it all together. All right, I love this. So let's think about this. A pathogen can be a bacterium, which is unicellular, which is singular for bacteria. A pathogen can be a bacterium, but not all bacteria are pathogens. This is a huge, 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 huge misconception about bacteria in our world. That oftentimes when someone says bacteria, they think, oh, that's bad. It's usually we should be like, yay, bacteria, they're great. Most bacteria keep us healthy. Most bacteria are really, really important to our survival. It's those real few. Look at this, 1%, less than 1% of 1%. So very few bacteria are actually pathogenic. Most of them are really, really, really important to us. So if I put on the exam this statement and said true or false, you would say true. Bacteria are everywhere. I should say prokaryotes are everywhere. So if you've ever heard like kids who play in dirt are much healthier than kids who don't, that's because when you play outside in the dirt, you expose yourself to a lot more symbiotic bacteria. That the more you're exposed to, the more you add to your own body's microbiome. And we'll talk about that. It's the ecosystem that lives in and on you that's made of all kinds of microscopic things, which could also include archaea, bacteria, and we said there are some also protists. So all those little microorganisms, they help us to be healthy. So having exposure to the outdoors, we know just the microbiome aspect makes us healthier, but also psychologically, it also makes us healthier. The human body is home to trillions, and that's being very, very conservative, probably a lot more than that. We have bacteria all over our body. We have microorganisms all over our body. So let's talk about some of the archaea. The archaea are, whenever we say like really extreme, crazy environments, you're probably talking about an archaea prokaryote. Hot springs, they can live in boiling water or even near boiling points. If you hopped in a hot tub that was boiling, you would dissolve. You would begin to cook yourself. They can live in that. Down near the core of the earth. So this is an underwater picture. This is a hydrothermal vent. So down, down, down deep in the ocean, there are some little cracks in the core of the surface of the earth underwater. And from that little crack comes out stuff that's inside the core of the earth. The core of the earth is really, really, really hot. So you've got super duper duper hot temperatures. You've got all kinds of chemicals that are inside the core of the earth coming out. Those chemicals for us are toxic. So you've got organisms, you've got archaea who live down there. They're the core of these ecosystems way deep down in the ocean that are exposed to boiling temperatures and toxic chemicals. In addition, not only those things, but also water. So like a case of water is really heavy, right? So now let's think about like if you go down deep into the ocean. Some of these hydrothermal vents are thousands of meters down in the ocean. So, case of water. Pick up a case of water, pick it up, you're like, it's heavy. Imagine millions of cases of water on top of you. Would that crush you, the weight? Yeah. So water, even though when you go into water and you're like, this is nice, I'm kind of like weightless. But if you go deep underwater, even though you might not feel it, the pressure of that weight of water on top of you can start to actually crush you. So they can live in crushing, toxic, boiling environments. No sunlight either. So here we have bacteria that are the base of ecosystems. How the heck are there ecosystems where there's no sunlight? It's a totally different process from photosynthesis called chemiosynthesis. And they use these chemicals like photosynthesizers use sunlight. And those chemicals go through a process to make oxygen or whatever they need in terms of gases, but also make energy. So very different things here. Those cracks or fissures are called ocean vents, hydrothermal vents, hydro, water, thermal, hot, vent, crack in the core of the ocean. They also could be opposite direction in freezing things. If you go into cold water, you can get hypothermia. You can release all of your heat, and not having enough heat can cause everything to shut down, and we can die. But they can live in actual ice. It's impossible for most organisms. And then if we're talking about places that are very high in salt concentrations, like the Dead Sea, it's called the Dead Sea because most organisms cannot live in high salt concentration areas. Salt in general for cells, it's going to suck out the water. But you have communities that live there too. So usually in all these extreme, or what we call extremophile, they might call them extremophile bacteria, but they're not bacteria, they're prokaryotes called archaea. Okay, so let's talk about pathogens. Pathogens are organisms that cause disease. They cause disease by producing toxins that come out of their cells. Pathogens are part of the bacterial domain. So a few reminders. There's a lot of viruses and bacteria and other disease-causing things going around, especially this time of year. Remember 20 seconds hand washing. Rather than using very drying hand sanitizers, it is better for your body to just go in the bathroom and for 20 seconds wash your hands. It's also good to cover your face when you sneeze. Because when you sneeze, things shoot out in all directions except for it almost behind you, but it goes like this way too. It'll shoot out 20 feet in almost a circle around you if you don't cover your face. So that's just good, polite ways of protecting everyone else around you. All right, let's talk about a few examples. Clostridium botulinum causes botulism. They produce a toxin that paralyzes the cells that come in contact with the toxin. Paralyzes them. So that means those cells can't work. So some brilliant mind who worked in the pharmaceutical industry was like, hey, this toxin, it can paralyze cells. So what if, because people just they want to look young their whole life, what if we take that toxin and we inject it in little bits into areas where when you smile or make faces, you get wrinkles. And we inject it into the muscles in your face to cause them to be paralyzed so that when you smile, all the muscles are paralyzed and you don't get the lines. That's what Botox is made from. So somebody figured out the connection to make the muscles in your face from Botox cause you to not have wrinkles over time. They also use this to inject, like if somebody sweats a ton, what they'll do is they'll use Botox and they'll inject it into your armpits, and the armpits will get paralyzed so they don't produce sweat. And so somebody who's like a hyper sweater, it'll cause them to not sweat as much. There's other applications, more medical applications, rather than just freezing your face from wrinkles. A cousin of that is Clostridium tetani, so a different species of Clostridium bacteria. Tetanus can be found on rusty metals. So like if you're walking around, if you're in your yard and there is a nail and it's rusty and you step on it, you can get a wound, and then that Clostridium tetani, it will start to produce that paralyzing toxin. But the paralyzing toxin doesn't travel through the body, it just stays in that wound area. And so what it does is that wound area gets paralyzed, and so the immune system gets paralyzed from that area. Everything that comes to that area paralyzed, so the wound never heals. And so then what happens is the wound gets bigger and bigger and bigger and bigger over time. So every 10 years, there's a vaccine that you can get for tetanus. If you don't know when your last tetanus shot is, it's not a bad idea to figure that out. And every 10 years, you get it. Like me, I know on the 9s, every, like 2029 will be my next tetanus